I have a little animation to kind of help you understand the mechanism of both enzymes. So we'll start with transketolase, right? What happens? So always we start with ketose of the two. So we have a ketose and aldose reacting with each other. We'll cleave the bond in the ketose. So the bond will be cleaved between between the carbonyl carbon and alpha carbon, and the top portion of a ketose will be placed on top of the aldose to form a new CC bond, right? And of course, the remaining first carbon of ketose will be converted to an aldehyde, right? And of course, we kind of switch the notations, right? So the bottom portion of aldose becomes a ketose, and the bottom portion of ketose becomes an aldose. In case of transaldolase, we cleave the bond between alpha and beta carbon, but everything else is the same. We start with a ketose. The top free carbon fragment gets transferred on top of an aldose. The CC bond is going to be formed, and ketose is converted to aldose, right? Because here this carbon becomes aldehyde carbon. Okay, so now I have a couple of questions for you to check if you understood the material correctly. So here you have some options for the products of transketolase catalyzed reaction between fructose 6-phosphate and glucose 6-phosphate. So here you have those structures, fructose 6-phosphate and glucose 6-phosphate. Please pause to think as long as you want, and I will provide the correct answer in a second. So transketolase catalyzes transfer of two carbon fragments from a ketose on top of an aldose. So this is our ketose, right? Fructose 6-phosphate. Here are two top carbons. So we are going to cleave this bond. And so those two carbons are going to be added to that carbon, right? So as a result, the bottom portion of fructose 6-phosphate becomes an aldose, 4-carbon aldose, such as erythrose 4-phosphate shown here. And this top portion becomes the top portion of a new ketose, which would contain the original 6 carbons of glucose 6-phosphate plus those 2 carbons, 8 carbons total. So the correct answer will be option A. So now let's see what will happen if another enzyme, transaldolase, is used to catalyze the reaction between the same two monosaccharides, fructose 6-phosphate and glucose 6-phosphate. Again, I will give you some time to think. You can pause the video now, and I will provide my answer in a second. So transaldolase catalyzes cleavage of the CC bond between alpha and beta carbon of a ketose, right? And it would result in the transfer of free carbon fragment from this ketose on top of the second substrate, which is aldose. So as a result, the bottom portion of this ketose becomes an aldose. So we need to look for the 3-carbon aldose, such as glyceraldehyde 3-phosphate shown here. And the ketose that is going to be produced will contain those 6 carbon atoms of glucose 6-phosphate plus 3 carbons that we transferred from fructose 6-phosphate. So we are looking for 9-carbon ketose. So the correct option will be B. I hope you correctly identified the right products. So the only thing that I still want to talk about a little bit is 3 different modes of pentose phosphate pathway. So again, if those modes operate depending on the need of the cell. So if the cell needs to synthesize nucleotides, let's say it's rapidly divided cells. So the pathway, the whole pentose phosphate pathway needs to operate so that to produce NADPH, of course, but also ribose 5-phosphate. And so in this case, both oxidative phase and the first isomerization stage of the non-oxidative phase are in effect, because the cell needs both ribose 5-phosphate and NADPH. So what if we don't need to synthesize a lot of nucleotides, but anabolic pathways in general are in effect, right? So we require a lot of NADPH. So in this case, again, oxidative phase is needed because this is where NADPH is produced. And this ribose 5-phosphate, which is the product of the oxidative phase, needs to be metabolized all the way to synthesize glyceraldehyde 3-phosphate and fructose 6-phosphate. and they react with each other so that to form fructose 6-phosphate, of course, can be easily isomerized into glucose 6-phosphate, but glyceraldehyde 3-phosphate needs to react with dihydroxyacetone phosphate to form fructose 1,6-bisphosphatase. Again, that could give us fructose 6-phosphate and glucose 6-phosphate. And this glucose 6-phosphate is the substrate for the pathway, so it's gonna be reused, so it's gonna be cycled through the pathway to generate as much NADPH as possible. But in some intermediate cases, then neither nucleotide synthesis is required, nor other anabolic processes are very active. So in this case, of course, oxidative phase will operate to produce NADPH, but once we form fructose 6-phosphate and glyceraldehyde 3-phosphate as a result of non-oxidative phase, those two molecules enter glycolysis to supply the cell with ATP, right? Not to waste those carbon atoms that can donate electrons to further drive oxidative phosphorylation. So since glucose 6-phosphate is a starting compound for pentose phosphate pathway, that's why it's called pentose hexose monophosphate shunt, right? Because it shunts glucose 6-phosphate away from glycolysis. But what is the signal that tells the cell to switch between the two pathways? And in general, that's substrate availability. Availability of the substrate for the first step of the pentose phosphate pathway, such as oxidation of glucose 6-phosphate. So if we have high concentration of NADPH in the cell, we don't need to produce more. That's why most of glucose 6-phosphate is used for glycolysis for energy generating purposes. If concentration of NADPH drops, it means that NADP plus concentration gets increased, right? And NADP plus is the second substrate for glucose 6-phosphate dehydrogenase, and just by substrate availability, it activates the enzyme that activates the whole pathway in general. This is it for pentose phosphate pathway. Please email me if you have questions. Thank you for your attention.